So in this lecture, we're going to be covering mitosis and meiosis. So as we're soon going to see, these are two very important processes that some organisms and all cells go through. So chapter 8 is a very meaty chapter. So there are some topics I don't want you to focus much on. In particular, I don't want to focus on the cell culture material, cancer, or growth factors in the cell cycle. So I think these topics are very interesting, which is why I have included them in your reading. But for exam purposes, don't worry too much about those topics. So what's the point of mitosis and meiosis? So really there are three reasons: reproduction, growth, and development. So if you remember the slide from the first lecture we had, these are the 7 characteristics of life, and so mitosis and meiosis cover 2 of those. So again, reproduction, growth, and development. So here's that again, just written out for you. So for asexual reproduction and growth, cells and organisms use mitosis. So mitosis is all about creating identical cells. As you're going to soon see when we go over the process of mitosis, creating 2 identical cells. So asexual reproduction may be something you're not very familiar with. So think of a starfish. So a starfish can actually tear off its arm, and that arm can actually regrow an entire starfish. So the new starfish as well as the old starfish are actually clones of each other. So through the process of mitosis, creating identical cells, that arm has been able to regrow the entire starfish. On the other hand, we have sexual reproduction, and that's done through the process of meiosis. So meiosis is about creating sperm and egg. And as you all know, sperm and egg meet, and they create a new organism. So here's a little bit more information on mitosis and meiosis. So both of these processes are really just organized movement of DNA within the cell. So really the whole entire point of doing mitosis and meiosis is to make sure the cell has the right amount of DNA when it splits. So if there's something wrong where one cell gets more DNA or one cell gets less DNA, that's very bad for the organism. So mitosis and meiosis are all about making sure before a cell splits and makes a copy of itself, or it turns into a sperm or egg, that the cell has the right amount of DNA. And again, I already alluded to this, mitosis creates 2 daughter cells, and meiosis creates 4 daughter cells. So at the very end of mitosis, you have 2 cells. At the end of meiosis, you have 4 cells. So here's a quick reminder. So DNA, as you know, is stored in the nucleus of the cell. So this picture should actually look fairly familiar to you. So this is a cheek cell actually stained with methylene blue. So methylene blue, as you may remember from lab, actually stains DNA. So you can actually see the nucleus of the cheek cell, just that little blue dark circle in the middle of the cell. So the DNA found in the cell is actually stored in things called chromosomes. So here is your standard DNA double helix. It has those 2 backbones running along this length. And so DNA is actually wrapped around proteins called histones. So the DNA in your cell is not actually just DNA, it's DNA wrapped around these things called histones. And again, those are proteins. So as you can see, they form these bundle of 8 histones, and we call that a histone octamer. So octamer, octo of course means 8. So essentially you have DNA wrapped around these histone proteins. So this histone proteins actually allow DNA to be raveled. So as you can see, we start forming coils here. So DNA has a double helix, but now we're forming coils. We're sort of packing the DNA more and more. Eventually it forms things called supercoils, so even more coiled than coils. And eventually we pack the DNA so tightly that they form these things called chromosomes. So chromosomes, you can just think of them being very, very, very tightly packed strands of DNA. So I like to think of this being almost like strands of yarn. So if you just have a pile of yarn all tangled together, it's very, very hard to move. You have to sort of grab it and try to pick it all up at once. It's very challenging. However, if you wrap it nicely and pack everything very tight, you have things like these bundle of yarns right here on the left-hand side of this PowerPoint slide. So those, of course, are much easier to move. So these are like chromosomes. So chromosomes are tightly packed DNA. Here you have tightly packed yarn. So again, it's very easy to move compared to that bundle of yarn, or the pile of yarn, I should say, on the right-hand side of this PowerPoint slide. So this is essentially what the cell is doing, just packing its DNA up so that it can be moved more efficiently. And of course, mitosis and meiosis, again, is the coordinated movement of DNA, so you want DNA to be easy to move so you don't make any mistakes. So that is why the cell packs their DNA into chromosomes during these processes. So next up, we have karyotypes. So these are essentially a picture that show you what chromosomes an organism has. For a human, we would see 46 chromosomes on a karyotype for us. So we have 23 pairs of homologous chromosomes, which I'll define in just a second. So 23 pairs, so 23 essentially from dad, 23 from mom, to make a total of 46 chromosomes each. So homologous chromosomes are kind of a tough concept for students to understand, so I'll go slow through this definition. So homologous chromosomes have the same order of genes. So the homologous chromosomes, say chromosome 1, you have one chromosome 1 from dad, one chromosome 1 from mother. So here, those would be homologous chromosomes. You would have the same order of genes, just your genes from dad are probably going to be slightly different from the genes from mom. So you may have a different base pair, so maybe instead of a cytosine, you have an adenine, something like that. So they're the same genes, just slight variations between mom's version and dad's version. So homologous chromosomes, again, have the same order of genes, but that same gene can be slightly different between mom and dad. So hopefully the next picture, which is a karyotype of a human, will give you a better idea what I mean with that. So as you can see, we have 23 pairs, 22 autosomes. So autosomes are chromosomes that are not sex chromosomes. So autosomes, as in auto, A-U-T-O, S-O-M-E-S. Here, this is a male, so males are XY. And those are the sex chromosomes of this person. Females, I should say, are XX. I'm pretty sure you already know that. So males are XY, females are XX. So humans have 22 pairs of autosomes and 1 pair of sex chromosomes. So here you can actually see the homologous chromosomes. So here maybe we have one from mother, one from dad, one from mother, one from dad, so on and so on. So for each pair of homologous chromosomes, one comes from dad, one comes from mother. Again, they have the same order of genes, just the genes can be slightly different in variation. So they may have slightly different base pairs, but essentially they're going to code for the same protein. So here's a question for you to ponder. So question 1 for Blackboard. So an organism that has 40 chromosomes will have how many chromosomes in each cell after mitosis? So A20, B40, C80, or D160. So take some time to answer that. In a little bit, I'll come back and answer this question. So this question here is to remind you what the function of mitosis is, and that's to create 2 identical cells. So if you have an organism with 40 chromosomes, you want all of its cells to have 40 chromosomes as well. So at the end of mitosis, you'll have 2 cells, each with 40 chromosomes. So the correct answer here is B, 40 chromosomes. So next up, we're going to talk about the cell cycle. So the cell cycle I like to think of being like the clock for the cell. So the cell cycle tells the cell when it's time to start doing mitosis, when it's time to start synthesizing DNA, and when it's time to make a new copy of itself. So there are really 4 parts to the cell cycle. There's G1, S, G2, then M. So G1, S, and G2 are called interphase. So interphase is really all about preparing the cell to do mitosis. So you have things like checking the size of the cell, making sure there are enough organelles, making sure the DNA is duplicated, things like that. So as you can see, interphase actually takes up the most of the cell's time. So the cell spends most of its time in interphase. So G1 and G2 are really about checking to make sure the cell's the right size, there's enough organelles, and things like that. S phase is very important though. So S phase is actually where you have DNA replication happen. So S phase is very important to see if all the chromosomes duplicated in that phase. So as you can see, M phase is actually very short. So M phase is where we see mitosis happening. So mitosis is actually a very quick event compared to everything else. So again, really the point of the cell cycle is to control cell division or mitosis in the cell. Some cells also have a phase called G0. So G0 cells are actually cells that are not dividing. So a lot of cells like muscle cells and neurons, which don't divide very often, many of those cells enter G0 phase. So G0 is when the cell's not dividing, so it's not actually going to go through mitosis anymore. So it's actually exited the cell cycle. So before the cell can actually undergo mitosis, again, we have to have DNA replication. So really the first step in mitosis is replicating DNA. So here we have a chromosome. Again, you can see it's made up of DNA, just that DNA has been tightly wound around those histone proteins. So now it's very tightly packed. It's condensed into those nice bundle of yarns that are easy to move. So during chromosome duplication, we have sister chromatids formed. So the chromosome on the left and the chromosome on the right are exactly the same. That's why they're called sister chromatids. And as you can see, we now have 2 strands of DNA. So this is DNA replication right before mitosis. We have the formation of sister chromatids. Eventually, those sister chromatids will be pulled apart, and you'll have individual chromosomes again at the very end of mitosis. So here I want you to know what a sister chromatid is. So essentially, it's a replicated chromosome. So it does actually look like that. So it has one chromosome there, another chromosome there, and they're actually held together at this thing called the centromere. So it's almost like the center of the chromosome, or sister chromatids. So be aware of that. Sister chromatids are replicated chromosomes, and they're exact copies of each other. Okay, so now we're actually ready to do mitosis. So we've duplicated chromosomes, so we formed those sister chromatids, and we made sure the cell is big enough and has enough organelles to split, so we're not going to destroy the cell in the process, so we have enough resources to actually do mitosis. So in the next few slides, I'm actually going to show you what the steps of mitosis are. So the first step of mitosis is actually interphase. So depending on how you think of this is not really technically a step of mitosis, but here the textbook has listed it as the first step for this figure right here. As you can see in interphase, the nuclear envelope is still intact. In other words, the nucleus is still there. As we're going to see later in mitosis, that nuclear envelope actually breaks down and disappears. You can also see the DNA inside of it. So that DNA is now just that messy pile of yarn. It's not condensed yet into those nice compact chromosomes. So that's actually going to take place in prophase, the next step of mitosis. We're actually going to see that DNA be coiled up. You also see the centromeres in yellow. So really the function of the centromeres is to start pulling apart DNA. And we're going to see that again in later steps of mitosis. So I like this figure since it actually shows you what this looks like in a real cell. So we have fluorescent microscopy done. So here you can see the nucleus in blue. Red is actin, which really doesn't play much of a role in mitosis, but it's still there. So you can see sort of the boundaries of the cell. And then in yellow, you can see the centromeres and some of the microtubules that actually connect them. The second step of mitosis is prophase. So here we've actually started mitosis, so prophase is definitely a step of mitosis, unlike interphase. You can see those centromeres now have early mitotic spindles. So these are actually microtubules that are being formed, and those are going to try and pull apart DNA, as we're going to see in later steps. If you look at the nucleus, our chromosomes are now condensed, so that DNA has become wound around those histones and they've tightened themselves up. So now they are condensed, and you can see that we no longer have that messy pile of yarn. In prometaphase, we have connection of those chromosomes to those mitotic spindles. So now you can see those mitotic spindles have now connected to what is called the kinetochore. So again, the point of the centromeres and the mitotic spindles is to move DNA around. So it's of course very important that they're connected to the DNA via those kinetochores, or else they're going to have issues moving DNA around. So these are the first 3 steps of mitosis: interphase, prophase, and prometaphase. So here's the second and final part of mitosis. So in this step, we have metaphase, anaphase, telophase, and cytokinesis. So in metaphase, the sister chromatids actually line up on the metaphase plate. So this is a sort of imaginary line. It's not actually a thing in the cell, but all the sister chromatids line up horizontally on this plate. So you can see they all line up in the same orientation. So again, I said earlier, the mitotic spindle is really what's doing this. So you can imagine the mitotic spindles are sort of like ropes or pulleys. So they're pulling the sister chromatids into place. In this case, they're pulling them into place on the metaphase plate. In anaphase, we actually have sister chromatids pulled apart. So you can think of anaphase. Anaphase starts with an A. Apart also starts with an A. So you can think anaphase, apart. So you can see those mitotic spindles are pulling those sister chromatids apart, and now they're being pulled to either side of the cell. In telophase and cytokinesis, we start having the nuclear envelope come back. So you can see sort of the early start of that, which I've just circled. Also, the centromeres start going away, since you don't need those anymore. And eventually we're going to have cytokinesis. So cytokinesis is actually where you split the cell apart. So these are the major steps of mitosis. So interphase, prophase, prometaphase, metaphase, anaphase, telophase, and eventually cytokinesis. So here's a little bit more detail about cytokinesis. So again, that's the event where the cell essentially pinches off from each other. So you have 2 daughter cells formed at the very end of mitosis. So it does actually look just like your textbook figure. So you actually have what's called a contractile ring that forms around or in the middle of that cell and squeezes it into 2 parts. It's important to note that animal cells, like the one right here, have a different type of cytokinesis than plant cells. So plant cells actually have a cell wall, which creates lots of issues, so they can't really do this pinching. So the next slide, I'll show you what it looks like in a plant cell. So in a plant cell, you actually have a cell plate formed. So plant cells go through all the same steps of mitosis as animal cells, just their cytokinesis is a little bit different because of the cell wall. So this cell plate that's formed essentially cuts the cell in half. So eventually it just keeps on growing and growing, and eventually it splits the cell in half, and you now have 2 daughter cells of the plant. So just be aware of that. Animal cells have this contractile ring, and they form what's called a cleavage furrow, and they eventually split into 2. Here, a plant cell has this cell plate, and eventually you have 2 daughter cells formed for that plant. So after all of that, we finally completed mitosis, and now we have 2 identical daughter cells. So remember, the point of mitosis is to do asexual reproduction, essentially clone yourself, or to grow. So I'm actually going to show you a video that goes through all the steps of mitosis. It's got a nice animation that comes from the textbook publisher, and I think it's going to do a good job of showing you what mitosis actually looks like.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of 2 identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
So hopefully you found that video very helpful. So here is now a question for you to answer on Blackboard. So I want you to identify the step of mitosis that is shown in the picture on the right. So here we actually have metaphase. So remember in metaphase, the sister chromatids actually line up on that metaphase plate. So it's sort of imaginary line running down the cell. So here the correct answer is B, metaphase. So here's just a quick review of mitosis. So the major idea here is we're trying to replace cells, grow, or do asexual reproduction. So that's the major purposes of mitosis. Two cells are produced, and each of those cells is identical. So if you started with a cell with 10 chromosomes, you would end up with two daughter cells, each with 10 chromosomes. So here's sort of a big overview of what meiosis is. So meiosis is all about producing sperm and eggs. So through the process of meiosis, as you can see here, we have egg cells and sperm cells produced. They then fertilize, and now we have mitosis used for development and growth. So here we have a human life cycle. So we have sperm and egg meet that forms a human, which goes through mitosis, and we form an adult at the very end of this process, and everything starts all over again. So we're going to see life cycles come up later in this course when we talk about biodiversity, but here's just a simple human life cycle for you to be aware of for this lecture. So as you may notice, there's this key in the top right-hand corner that talks about haploid and diploid cells. So the next slide will talk about what exactly it means by diploid and haploid. So for something that's diploid, that means we have two sets of chromosomes. So remember I talked about homologous chromosomes a little bit earlier in this lecture. So you had one set come from dad, one set come from mother. More specifically 23 from dad, 23 from mother, to make 46 chromosomes total. So since we have one coming from dad, one coming from mother, we have two sets of chromosomes, which makes us diploid. For sperm and egg, though, we want half as much. So we want one complete set of chromosomes. And that is what we call haploid. So haploid, you can think of half. So you want half the number. And of course, the math works out to be 1 plus 1 equals 2. So you can imagine one sperm cell plus one egg cell equals two sets, or two complete sets of chromosomes. So essentially we have one complete human. So we don't want to have 2 complete sets in a sperm cell and 2 complete sets in an egg cell, because if those meet, then you would have 4 complete sets and you would have twice as many chromosomes as you need in a human. So here we want to split the number of chromosomes in half, so we want to be haploid, because we want half the number so that when sperm and egg meet, they create a full set of human chromosomes. There's also some common abbreviations. So CHU-N means diploid, and N by itself means haploid. So just be aware of that. You may see that somewhere in the textbook. So CHU-N means diploid, N means haploid. So sometimes we also see CHU-N equals 46. So that just means a diploid organism. So the CHU-N organism or diploid organism has 46 chromosomes, as in humans. You can do some simple algebra. So 46 divided by 2 equals 23. So here a haploid sperm or egg has 23 chromosomes. So here's a nice slide that just goes over what I've talked about. So in mitosis, remember, we're creating identical cells. So CHU identical daughter cells. So here we start off with a diploid cell with 46 chromosomes, and we end up with 2 daughter cells that are also diploid as well. So they have 46 chromosomes. In meiosis, though, it's different. You start off with a diploid cell with 46 chromosomes, and at the end of meiosis, you have 4 cells, each with 23 chromosomes. And remember the word for that, since the CHU-N equals 46, and the N equals 23, here we have 4 haploid cells formed during meiosis. So in mitosis, we have 2 diploid cells formed. In meiosis, we have 4 haploid cells formed. Haploid, remember, think of the half, so you have half the number of chromosomes in the meiosis daughter cells. So now we're going to go over the steps of meiosis. So as you're soon going to see, meiosis is very similar to mitosis. So I'm really not going to go into excruciating detail about every single step, but I'll sort of point you to the major differences between mitosis and meiosis. So the first step in meiosis is interphase, just like mitosis. So we have duplication of DNA in that step. You can see the nuclear envelope is still there, just like mitosis interphase. And we also have the centrosomes formed as well, so that can pull apart DNA later in the process of meiosis. In prophase 1 of meiosis, this is a little bit different from prophase and mitosis. So I'm going to point out some very important features. So the first thing is these tetrads that are formed. So essentially these are formed by homologous pairs of chromosomes, or I should say homologous pairs of sister chromatids at this point. So essentially you have homologous sister chromatids laying on top of each other. So essentially you have 4 chromosomes total, which is why it gets tetrad, so tetra means 4. So essentially you have homologous sister chromatids sitting on top of each other. And this is going to allow for the process of crossing over. So a large part of meiosis is about creating genetic variability. As we're going to see in evolution and ecology, genetic variability is actually very helpful for organisms. So meiosis actually doing this crossing over event, where it exchanges DNA between homologous sister chromatids, is actually very beneficial for the organism since it's creating variability. So essentially you can see that for something like this tetrad here, we have DNA exchange between the blue sister chromatids and the red sister chromatids. So essentially we're sort of swapping a little bit of DNA between these sister chromatids, creating more variation there. So that's the act of crossing over. Other than these tetrads forming and this crossing over, there's really not anything else majorly different between prophase 1 and prophase of mitosis. Metaphase 1 of meiosis is actually very different from metaphase of mitosis. So here, instead of sister chromatids lining up one by one on the metaphase plate, here we have homologous chromosomes lining up. So now we're not actually going to have sister chromatids pulled apart, we're going to have homologous pairs pulled apart. But just like metaphase of mitosis, here in metaphase 1 of meiosis, they're lining up on the metaphase plate. Just not in a single file row, but here we have homologous pairs lined up next to each other. Anaphase 1, again, since we don't have a single file row of sister chromatids lined up, here instead of separating sister chromatids, we're separating homologous pairs in anaphase 1. They could pull to other sides of the cell. And then we have telophase 1 and cytokinesis 1. So essentially, CHU cells are formed at the end of meiosis 1. So in prophase 2, this is actually very similar to prophase of mitosis. So there's no more crossing over and no more tetrads formed. We're just having the mitotic spindles come back and getting ready to do their job. As you can see, the nuclear envelope is still gone as well. In metaphase 2, exactly the same idea as metaphase of mitosis. Here you're splitting sister chromatids. So now they're lining up in that single file row. You have splitting of sister chromatids as opposed to metaphase 1 of meiosis, where you have splitting of homologous pairs. Anaphase 2 is exactly the same as anaphase of mitosis. Yes, splitting apart or pulling apart of those sister chromatids. And eventually you have telophase 2 and cytokinesis, and you have the formation now of 4 sperm cells or 4 egg cells, each of them being haploid cells. So this is meiosis. So major differences, you have crossing over, tetrads formed. Of course, you also have essentially 2 sets of this process. You have meiosis 1, meiosis 2. But very similar idea to mitosis, a lot of similar steps and similar concepts. So here's the final question for Blackboard, so question number 3. So what step of meiosis is shown in the picture on the right? So here you can see the sister chromatids are lined up on the metaphase plate. So here you can clearly see the sister chromatids are lined up on the metaphase plate. And you can imagine in this step, or the next step that's going to come up in anaphase, we're actually going to have the sister chromatids pulled apart. So in meiosis, when you see the sister chromatids lined up one by one like this is metaphase 2. In metaphase 1, you have homologous pairs lined up. In this case, we have sister chromatids lined up instead. So here, this is actually metaphase 2, so the correct answer here is D. So we talked a little bit about this. So creating variation is very important for meiosis. So one of the ways we can do this is crossing over, which is shown on the left. So here you have paternal and maternal sister chromatids forming tetrads, and you have genetic material exchange between those two. So that is crossing over. The second way in which we can have variation created is through independent assortment, which is shown on the right. So essentially this just says that you can have homologous pairs line up differently on the metaphase plate during metaphase 1 of meiosis. So here we have one blue on the left, one blue on the right, one pink on the right, one pink on the left. So that's one possibility, or one way in which these chromosomes can assort on the metaphase plate. The second way is we can have CHU on the left and CHU on the right. So that's the second way in which we can assort chromosomes during metaphase 1 of meiosis. So depending on this independent assortment of chromosomes in metaphase 1, this is going to change the outcomes in later steps of meiosis. So as you can see at the very end, we essentially have 4 different possibilities due to independent assortment during metaphase 1. So here we're only dealing with 4 sister chromatids. As you can imagine, if you have 46 chromosomes as in humans, there's a lot more possibilities about how these chromosomes can assort during metaphase 1. And because of that, independent assortment allows for many, many different possibilities. So I'm going to leave you with this slide right here. So this is the comparison to mitosis and meiosis. So as we saw, they're very similar processes. So maybe a little later, maybe not today, but maybe in a week or two, go over this slide right here again, look at it, and see if you can figure out the differences between mitosis and meiosis. So as you saw, there are some major differences, but there are also a lot of similarities, so look out for those as well.